Okay, imagine this for a second. A tiny, tiny machine, I mean microscopic, built from DNA itself.
The blueprint of life, yeah.
Exactly. And this machine can actually move, it can perform tasks, almost like, the little motors inside our own cells.
It's pretty amazing stuff. And what's really fascinating is how scientists are trying to engineer these artificial motors, especially the DNA ones.
Trying to get them to work as well as the natural ones?
Precisely. Those motor proteins in our bodies are incredibly efficient. They're the benchmark, really.
And that right there is what we're diving into today. We've got some really groundbreaking research.
Right, this is exciting.
It's a study from Nature Communications titled Rational Engineering of DNA-Nanoparticle Motor with High Speed and Processivity Comparable to Motor Proteins.
It's peer-reviewed, solid science. A real leap forward, I think, in nanotechnology.
Definitely a leap. Now, we know keeping up with all this cutting-edge science can feel, well, overwhelming sometimes.
Like drinking from a fire hose.
Yeah. So think of this deep dive as your way to get the key insights, the exciting bits, without getting totally bogged down in the technical jargon.
We'll break it down.
Our mission today is pretty clear. How did these scientists manage to build a DNA motor that's fast and can keep going like the biological ones? And, why does it even matter?
Okay, let's start with the basics then. Molecular motors.
Right. We have the natural ones in us, and then these artificial ones scientists are building. What's the big picture?
Well, at the nanoscale, these motors are the workhorses. Inside our cells, you've got proteins like kinesin, constantly moving things around, making muscles work. They're fast, they're persistent.
Yeah, go the distance.
They do. Now, the artificial ones, especially DNA-based ones,
Yeah.
they offer amazing design flexibility. DNA is like Legos for nanotechnologists.
But historically, these artificial DNA motors have been, let's say, a bit disappointing in performance compared to nature's version.
Yeah, that's fair to say. Much slower. We're talking speeds maybe around 0.1 nanometers per second.
Yeah.
Tiny distances.
0.1, that's glacial.
It is, especially when you compare it to motor proteins zipping along at like 10 to 1,000 nanometers per second.
Right.
And they can travel over a micrometer.
1,000 nanometers. Huge difference.
A massive difference. And they don't just stop after a few steps, usually.
So obviously scientists have been trying hard to bridge that gap.
Oh, absolutely. There have been lots of attempts. People tried using RNA tracks on carbon nanotubes that gave long travel distances but still slow.
Okay.
Others used DNA on tiny particles moving on RNA surfaces. They got a bit more speed, but the motors tended to fall off easily.
Yeah.
Low endurance, low processivity.
Right. So this key challenge emerges, getting both high speed and that long run length, the high processivity at the same time, especially in these really tiny nanoscale motors.
Exactly. And that's where this new study by Harashima and colleagues comes in. They took a fresh approach.
What did they do differently?
They used a gold nanoparticle, about 100 nanometers wide, coated with DNA strands. And this moves along a surface coated with RNA strands.
Okay, DNA particle, RNA surface. What makes it good? What's the fuel?
The fuel, or maybe the catalyst, is an enzyme called ribonuclease H, RNase H for short.
RNase H sounds important. How does it drive the movement?
Okay, so the mechanism is called a burnt bridge Brownian ratchet, BBR. It sounds complicated, but the idea is quite neat.
Lay it on me.
Think of the DNA on the particle and the RNA on the surface as being complementary, like puzzle pieces. They can stick together, forming a DNA double strand or duplex.
Okay, they bind.
Right. Now, RNase H comes along, and its specific job is to find these DNA duplexes and cut up the RNA part.
Ah, like molecular scissors snipping the RNA track.
Exactly like that.
Yeah.
So once the RNA at a particular spot is cut, the DNA on the particle can't bind there anymore. It's like a bridge that's just been burnt behind it.
I get it. So it can't easily go backwards to where it just was.
Precisely. The random thermal jiggling Brownian motion still pushes it around, but the burnt bridge makes it much more likely to move forward onto the next available RNA segment. It ratchets forward.
That's clever. Using an enzyme to create directionality. So how do they actually watch this happen? It's incredibly small.
Yeah, they needed some serious tech. They used high-speed, high-precision, single-particle tracking. Think super-fast video microscopy.
How fast?
Up to 1,000 frames per second. Much faster than previous studies. This let them see the individual movements, not just the average drift.
1,000 frames a second. So what did that detailed view show them? How was the motor actually moving?
It confirmed the movement wasn't smooth. It was more like pauses and then sudden steps. Like it would sit there for a bit, then jump forward.
Pauses and steps. Okay. What did they find when they started with, say, low amounts of the RNase H enzyme?
Right. At low concentrations, around 36 nanomolar, the pauses were really long, like 70 seconds on average between steps.
70 seconds. Wow.
Yeah, an eternity at that scale. The step size itself was pretty consistent, around 20 nanometers, but those long pauses meant the overall speed was slow, about 0.33 nanometers per second.
So the pauses were clearly the bottleneck there.
Absolutely, that was the main limiting factor.
Okay, so the obvious next step. What happens if you crank up the RNase H? Add a lot more enzyme.
And this is where it got really interesting. They increased the RNase H concentration massively, up to 7,200 nanomolar,
Okay, a big jump.
and the pause times just plummeted, down to an average of only 0.2 seconds.
0.2 seconds. From 70, that's huge.
Dramatic reduction.
Yeah.
And crucially, the step size, that 20 nanometer jump, stayed pretty much the same.
So shorter pauses, same step size. The speed must have just shot up, right?
It absolutely did.
That's
At those high RNase H levels, the motor speed hit around 100 nanometers per second.
100. That's right in the range of natural motor proteins.
Exactly. That was the big headline finding. They achieved protein-like speeds.
Amazing. So they cracked it. We have super fast DNA motors now.
Well, almost.
Yeah.
There was a trade-off, a significant one.
Of course. What was it?
While the speed went way up, other performance metrics went down. The processivity, basically how many steps it could take before falling off, decreased quite a bit.
So it was fast, but didn't last as long.
Right. And the total distance it traveled, the run length, also got shorter. And it wasn't quite as good at moving consistently in one direction. Its unidirectionality decreased.
Okay, the classic speed versus endurance problem. It's fast, but maybe burns out quicker or gets lost more easily.
Something like that, yeah. High speed came at the cost of stamina and straight-line travel.
So to really understand why this trade-off was happening, they turned to computer modeling.
Exactly. They built a sophisticated simulation, a geometry-based kinetic model. It tried to capture everything happening at the molecular level.
Like the DNA binding, the enzyme attaching, the RNA getting cut.
All of it, at individual binding sites on the surface. A very detailed picture.
And did the simulation match what they saw in the real experiments?
Remarkably well. It reproduced the stepping motion, the pauses, and critically how everything changed as they increased the RNase H concentration. It matched the experimental data beautifully.
That's powerful. When your model reflects reality that well, what key insight did the simulation give them about this speed-endurance trade-off?
It pinpointed the changing bottleneck. At low RNase H, the simulation confirmed the slowest step was waiting for an RNase H enzyme to arrive and bind.
Okay, the enzyme finding the spot.
But as they flooded the system with RNase H, that step got faster and faster until it wasn't the bottleneck anymore. Instead, the limiting step became the DNA on the nanoparticle finding and hybridizing or binding to the next available RNA site on the surface.
Ah, I see. So with tons of enzyme cutting away the track super fast, the motor's own ability to grab the next handhold becomes the limiting factor.
You got it. The enzyme is working so fast clearing the path that the motor struggles to attach downstream quickly enough to maintain a long, continuous run. This leads to detachment or wandering off course more often.
And that explains the drop in processivity and run length.
Precisely. The simulation also helped them classify the BBR mechanism. At low RNase H, it's closer to a perfect ratchet where things are well coordinated. At high RNase H, it becomes more imperfect because the cutting might get ahead of the binding.
Okay, so they've diagnosed the problem using the simulation. The binding speed needs to keep up with the cutting speed. So how do you engineer a solution?
Well, the simulation didn't just identify the problem, it suggested a solution. It predicted that if they could increase the rate of that DNR-RNA hybridization, make the motor grab the next RNA site faster, they might improve the trade-off.
Makes perfect sense. Boost the binding speed to match the high enzyme activity. How do they actually do that? You can't just tell the DNA to hurry up, can you?
Not quite. But you can redesign the DNA sequence itself. They engineered a new DNA sequence for the nanoparticle.
How? What did they change?
They introduced repetitive sequences and increased the amount of guanine G and cytosine C base pairs. G pairs form stronger bonds than adenine A and thimerosal TU, and sequence design can influence binding kinetics.
Okay, so they tweak the DNA code to make it stickier or faster at finding its match.
Essentially, yes.
Yeah.
They did separate experiments just to measure this and confirmed their new engineered sequence hybridized almost 4 times faster, 3.8 times to be exact, than the original one.
Wow, a nearly 4-fold increase in binding speed. That's significant. Did it work? Did this Mark II motor perform better?
It really did. This was the payoff.
Mm.
They tested the engineered motor, again at that high RNase H concentration where the original motor struggled with endurance.
Yeah.
And the results were impressive. It achieved simultaneously a speed of 30 nanometers per second,
Still pretty fast.
a processivity of 200 steps,
Much better than before at high speed.
and a run length of 3 micrometers.
3 micrometers, that's a really respectable distance at the nanoscale. That sounds genuinely comparable to natural motor proteins now.
It absolutely is. They hit that sweet spot, decent speed, good processivity, and a long run length, all working together. It showed that understanding the mechanism allowed them to rationally engineer a better motor.
That's fantastic. It's not just trial and error. It's targeted design based on understanding. So, stepping back, what are the broader implications here? Why is achieving this protein-like performance in an artificial motor such a big deal?
Well, on one hand, it helps us understand the design principles of nature's motors better. By building mimics, we learn about the original.
Right?
But maybe more excitingly, it really opens the door for practical applications. Having artificial motors that are fast and persistent, that's kind of the dream for nanoscale machinery.
What kind of applications are we talking about? Down the road, obviously.
Think about things like ultra-sensitive diagnostic tools, where motors could actively search for specific molecules, or molecular assembly lines, building complex structures atom by atom.
Or maybe even targeted drug delivery, having little motors that can actually travel effectively inside the body.
That's definitely one of the long-term visions, yes. Delivering drugs right where they're needed, potentially with speeds that match biological processes. This work lays important groundwork for that kind of future tech.
Did the study mention anything else interesting? What about the size of the nanoparticle itself? Does that matter?
They did look at that with their simulations.
Yeah.
They vary the particle size from 100 up to 5,000 nanometers.
Bigger is faster.
Surprisingly, no. The simulations suggested size didn't have a major impact on speed within that range. A larger particle might take slightly bigger steps, but it also seems to pause for longer between steps.
So it cancels out.
Pretty much, according to their model. The diffusion time increases, balancing the larger step size. Interesting finding.
Definitely. What about remaining challenges? Where does this research go next?
They do acknowledge limitations. One issue is still detachment, especially getting trapped in areas where the RNA track is already heavily degraded, the burnt bridges. This still limits the ultimate run length.
So how could they fix that?
They suggest a couple of routes, maybe using micropatterned surfaces, creating defined lanes or tracks for the motors to follow, or using non-spherical nanoparticles.
Like what?
Things made with DNA origami, perhaps, or gold nanorods. These have defined shapes and could potentially move more directionally, resisting getting stuck or wandering off track.
More control over direction and preventing detachment. Makes sense.
So yeah, the journey isn't over, but this study provides a really strong foundation and a clear strategy. Understand the kinetics, find the bottleneck, and engineer the components to overcome it.
It really highlights the power of combining experiment, high-resolution observation, and simulation.
Absolutely. That synergy was key to this breakthrough.
Okay, so for you listening, let's wrap this up. The really big takeaway here, the aha moment, is that scientists have managed to engineer a DNA motor that moves with the speed and, crucially, the endurance approaching that of natural motors.
And they didn't just stumble upon it.
No, exactly. They did it by digging deep into how it worked, identifying that crucial bottleneck, first the enzyme binding, then the DNA-RNA binding, and then rationally designing a modified DNA sequence to overcome it. It was smart mechanism-based engineering.
And why this matters is, well, it's a big step for nanotechnology, for synthetic biology. It shows we're getting much better at building functional machines at the molecular scale.
And it also gives us a deeper appreciation for just how elegantly nature solved these problems billions of years ago.
Definitely.
So maybe the intriguing thought to leave you with is this. Now that we can build these high-performance nanoscale motors, what incredible things could they actually do in the future once we get even better at steering them and keeping them on task?
Yeah, the possibilities for sensing, computation, assembly, medicine, it really sparks the imagination.
It really does. If your curiosity is piqued and you want to go deeper, we definitely recommend checking out the original paper. It's in Nature Communications, and we'll put a link in the show notes.
Or just searching for terms like DNA nanotechnology or molecular motors will open up a fascinating world online.
There's always more to explore.
Always.
Well, thank you for taking this deep dive with us today. It's a truly exciting field, this nanoscale engineering.
It really is. Looking forward to seeing what comes next.
Me too. Thanks for listening.